Welcome to follow the first podcast of Scaffdex and RegJoint anniversary year 2026. We celebrate this year with series of webinars and podcasts. My name is Tuija Annala and I am the founder and owner of Scaffdex. I have a pleasure to introduce the first podcast guest who is representing scientific background and owns extensive experience of research of bioabsorbable surgical implants, tissue engineering and regenerative medicine. Professor Minna Kellomäki from Tampere University Faculty of Medicine and Health Technology, Professor of Biomaterials and Tissue Engineering and Vice Dean of Research. Welcome.
Thanks, Tuija, for inviting me here.
Professor Kellomäki, we are now highlighting where this field in Tampere region and in general started a long time ago. Could you tell how you ended up to research of biomaterials? What kind of steps you took in studies and in your career?
I nowadays come from Tampere University, but it used to be Tampere University of Technology. So after high school and after having some 4 years in between, I ended up to study in Tampere University of Technology then, many years ago. And I think we knew each other already at those days, although we didn't then know that we are going to do like this type of scaffold works later together. I didn't plan to study technology when I was young, but I somehow ended up there. And then I really got interested in material science, especially soft materials and then also composite materials. So I wasn't fancying any steels or ceramics, but more of the plastic type of materials. And basically my medical biomaterial career started from the master's thesis. I got the good title or good subject for the master's thesis from that time the department leader or institute leader, Professor Pertti Törmälä, and I did my reactive extrusion polymerization of PLA work, and then I ended up doing next projects, and that's how it started. These fibers and scaffolds, they came actually kind of by accident as well. There was a colleague who was for a couple of years doing fiber spinning at the institute, and yeah, the story is a bit longer, so do you want to ask something in between?
As you told, history of surgical biomaterials is very long here in Tampere region. It is extended over several decades. Do you know how it was initially started here in Tampere? Who and when the biomaterial research started at the Tampere University of Technology?
That is also quite a good question. I last summer did a history presentation about that, and it's so long time ago that you don't find much by Googling. So you basically have to know where to dig out, and then you can find. It was Pertti Törmälä who did the technical part of that. So he was doing this polymer processing and modifying and functionalizing and all that kind of things. So they did first different kinds of plates and screws, and it's the so-called self-reinforcing. And he had a good colleague, Pentti Rokkanen, who was also originally working in Tampere, but then moved to University of Helsinki, and he was the clinician. So they were doing that work together. The other one was doing the technical side, and then the other one was testing the materials. So that was the start, and it was in the, I think, late 1980s. when they started to do, and those were actually the first um, like mechanically good small screws and plates for bone fixation, and they did a lot of like larger implants as well and things, but then later came these fibrous materials.
Concerning these fibrous materials, textile technology has a strong effect and purpose when these are produced and developed further. So what were the first target indications or ideas how these fibrous new materials could be used in patients?
The fibers were used as sutures to start with, and then came commercial versions of fleeces, and they were used for that they suck the blood from the like soft tissue in the surgeries, and also from there came that they were also biodegradable. And those were actually origin for that some rheumatoid surgeons started to fold those with their little own hands to small pillows, and then they stitched them a bit, and then they used them to the rheumatoid arthritic hands, to the small joints. And that was the starting point for these like joint scaffold type of implants. And then later, well, that was my colleague who did this before me. She started to think that, yeah, but these are having corners, and bones are not like cross-sectionally having corners. So how could we get rid of these corners? And then came the round and rolled structure. Of course, there were plenty of other things to do at the same time, but it was quite early when the round shape came. And why these surgeons came to ask help was that those materials, what they used, they were quite nice and porous, and they allowed the tissue to grow in. But then, on the other hand, they were degrading so fast that the nice space what was created between the bone ends was kind of destroyed because the tissue wasn't growing there like fast enough, and the pillow wasn't there for long enough time.
Okay, that's very interesting. You told that your colleague was participating or your before you in this development of this new structure. When did you step in the project, and what was your role in the beginning of this development work?
That's a good question. Now I have to think about the schedule. I think I came to picture by that I was helping her to do the spinning experiment. So we did melt spinning together, and that was already somewhere in 1993, 94. And then I was a while in London, and when I came back, we were for some time together at the same time, working in the research group, and then she went to do some other tasks in the companies, and then I ended kind of to inherit the project, so quite early.
Okay. And had you already your master thesis done in that phase? Was this before graduation or after graduation?
I had my master's thesis done at that time, and basically I was doing my PhD thesis, which at that time in Finland was taking some years. But yeah, basic things about fiber spinning and how the extruder is influencing to degradation of the fibers later. All those things were in my thesis at that time, of course. lots of things have been done later as well, but the initial data was there.
So that was the development phase of this new innovation and research of that. However, then something else is also needed if and when new medical innovations are taken to real clinical use. So typically regulatory affairs and authority approvals are essential if and when commercial devices are targeted. And in your faculty has been a source for several startup companies, as well as it is participating in research work of the companies. Can you tell about the phases from the innovation up to regulatory approvals? What kind of challenges were present when this kind of new joint spacer was developed, and how these challenges were solved? What was the time period? How long did it take until you could consider that maybe later on this could be a commercial product? Was it 1 year project or 3 years project or 5-year project?
Probably even more than 5 years, because there were first these kind of technical issues, and even though we were having interested in or keen surgeons who said that they could try those first ready-made scaffolds, which were of course sterilized, and all these things were also examined. It still took some time that they got the ethical approvals for those first preliminary experiments. And that was basically like one clinic study first, and with very few number, low number of patients, because they wanted to see, the concept was new, and they wanted to see that that's not harming the hands. And first patients were such that they kind of didn't have any other choices. The rheumatoid had destroyed the hands so much, and they had already had different type of implant there that had even more destroyed the bones. So they were challenging patients, and yes, the surgeons always went the patient ahead, so they wanted to see that it's functioning. So that also created a time span to the studies, because they wanted to have longer period of follow-up for the hands. But they quite soon, at the same time, they of course also tested the surgical techniques and stuff. But then when they were more convinced about that how that is going to work, then came a study where there were 2 or 3 clinics, and then came EU project, where also came along the company that was interested in that work and also gave support to these regulatory issues, and also like supportive help for that how these type of studies could be translated into products. So that was a long journey, and separate projects in that sense, that number of clinics that were participating, those were, they came more of the clinics. And of course before that and during that process, we needed to set up or set in stone some of the things, that all the scaffolds were as similar as possible, and these kind of things that typically are not for the university studies. So there were plenty of things done in that perspective. And of course, we are not experts in regulatory things, so it was good to have help.
If I recall it correctly, there were even several master theses done during that development work, when the clinical trials were already running. Can you highlight what kind of topics were handled by that manner, that what were the topics for those master theses, and what was the focus on those?
One student was doing that kind of work where she found from the literature how strong the hand can be when there is rheumatoid arthritic or osteoarthritic hand in question, and how that matches to the strength of the scaffolds. And of course, we needed also to set up how to test these scaffolds by, for example, compressing them, and how many parallel samples we need that we can be sure of that the scaffolds are equal enough when we say that they are similar. So those kind of things. But then also there were students that did some studies for the clinical side and also for the preclinical side. So these were tested also in the rats and in the pigs and these kind of things, because we needed to test that they are good enough and they are not harming.
So during the research phase at university, there were several different parallel projects ongoing, clinical trials as well as development of the production methods, testing of the implants, and the concept in technical side. However, there were quite intensive and big animal studies as well. So could you tell a little bit more about those most important animal studies?
There was a need to do the animal experiments as well, because they were needed for the registration if we were going to do them for the products later. And that was the reason that the things were running parallel. The animal experiments were running at the same time than the clinical, larger multi-clinical study. And indeed, Dr. Eero Varis was the person who planned the pig experiments, and there were the specific finger or toe of the pig that was chosen to be operated, and that was the one that didn't carry the load so much, so it was the outside finger, so to say, or toe. They planned it so that not only the function of the toe was analyzed, but also the histology of the space, the synovial space was studied. So those were really well planned. The people were calculating the statistics, how many animals are needed for that if they are showing this and that, then we can say that this is statistically significant or not. And there was also compared that time the so-called golden standard implant for those type of the works. And yes, there were lots of those master's theses from technical side, and for example, the sterilization of those scaffolds was one thing. On top of those ones, what I said, and those were also already going to direction again that if this one day is a commercial product, these could be used to show that these are sterile, and then also that the sterilization is lasting long enough time when they are stored for 1 month or half a year or 1 year. So I guess those were also in a way helping in the beginning to show that we can, or someone can sell them with that they can be used after 2 years of manufacturing. So different kinds of things. We had clever people in the team.
So that was the history and how this specific product was developed in collaboration at the university. But how about nowadays? What are the research topics and materials which are currently actively investigated by the university. These polylactide polymers have existed and used in surgical applications for 3 to 4 decades. So I'm sure that currently activities at the university are targeted more to some newer material options and applications. Can you tell about those?
Yes, we are, or we have been some 10 plus years been studying even more soft materials, like softer materials called hydrogels. So with those materials, not only us but others are trying to mimic the tissue itself, soft tissue namely, but of course we can always argue if they can be used also for hard tissue. So we are trying to study hyaluronic acid and collagen, fibrogen, just to name something. Most of these are originated from the nature, from tissue or from sea animals or seaweed, for example, or plants, other plants, or they can be made by bacteria so that the raw material comes from the production of bacteria. But that's the main thing, not only for us but for many others that are studying soft materials. Then on the other hand, I have a colleague who came already some years ago to work in our university. He's studying bioactive glasses, which also have been studied for decades. But since he's doing also synthesis of those ones, then he can swap their different ions and try to modulate the degradation and bioactivity of those glasses, and then study how those perform. And together we are doing composites out of these ones. Every now and then we go back to polylactides as well when we see something interesting and test those ones.
And if we think the past 15 years when this whole field has been growing and progressing, if you think about your own career, which have been the most important achievements and turning points during these past 15 years? Can you name those?
Things that have been kind of turning my career, I haven't, in a way, ever foreseen them. My theory is that I have been drifting from a challenge or opportunity to other. So I have been doing different things, and then I have been there like, looks interesting. Why not? What about if I try that one? And then, of course, one thing that has been very like driving to somewhere is funding, and having that Center of Excellence funding from Academy of Finland, and together with colleagues who are working from other fields, but we have had the same goal. Those have been good things for the new directions and also for the career in that sense. Maybe it can be like crystallized into that I have tried to take the opportunity. I have had nice and collaborative colleagues, and then there has been a possibility to get funding for those things that have been planned together.
If the hydrogels and bioglass are the present, what is then the future? What is your vision? What kind of new developments we'll see in future? For example, can artificial intelligence make work in laboratory completely unnecessary, or how this science science is developing further? What are the topics for next 15 years? Will we finally have this printer which is printing new tissues and cartilage for us?
That's always a good question. um, let's put it that way, that there is much hype around artificial intelligence, but I can see that somehow AI can help us, but not that way that we can get rid of everything, because we are not having good enough models for that they could predict the results for us. And I still think that we need to see that the new materials, for example, are working inside a body, and the cells can work with those ones. So how the AI will influence, I don't have that crystal ball, but it will somehow do that. In the meantime, we also need to remember that there are things that we want to bring into clinics and to like really help the patients, and that requires a lot of studies before that. And where are we working? Is that in the very early stages, putting different molecules together? That's a completely different work than that we are close to that it could be a product for a patient. So there is so much work that what can I say? I guess that some things that we are doing now in the lab and are new, they might be the new products in 5 or 10 years, maybe more to say 10 years. And those ones that we are just dreaming now, that what if I have funding or this for this idea, they will be in publications after maybe 1 or 2 years if we have that funding. But I guess that mimicking the body would be even more than what it used to be. So figuring out how the cells are working and how we can target the materials and, for example, drug release, that would be the thing in the future. We have more background knowledge now than what was known in mid 90s, for example.
That's interesting. I also believe that artificial intelligence can be a good tool, but biology is making things more difficult. It's not you cannot solve all the issues with biology with a computer. That's clear. Surprises will come.
Definitely surprises will come, and biology manages to surprise us every time.
What do you think of future of this field? Do we see tissue engineering changing the treatment of the patients within next few decades? What do you think?
Good question. And this is always good question, because we may have nice innovations and we may have nice products, but it's still up to clinicians. So doctors, surgeons, whatever we are speaking about, like specifically their professional title, it's up to them whether they see that it's useful and whether that is so much better than the current existing things that they are doing. So far, I think I haven't seen such thing that would really kind of wipe off all the others from that field.
If you can give one advice to the doctors, surgeons, researchers, and startup companies which are now entering into this field, what would it be?
One advice only.
Maybe 2, 3.
One is that check the history. check what is done earlier, and that means that not only last 5 years, but also a bit further back, what people have been doing, and have they been successful, or have they been ahead of their time so that it couldn't succeed at that time, or was it completely rubbish, and try to pick up those things that could be developed further. That's one thing.
And then when we think about what our discussion here during this podcast, what topic you would like to point out? What is the most important thing you would like to be remembered by the followers?
Trust yourself. And with that meaning that you may, when you're doing research, and also if you are commercializing things, you have the people that say that, yeah, well, that's not so good. And then there are those ones that say, yes, that's really, really something. And you may end up having like more of those suspicious things that, are you sure that that's going to work? Maybe not, maybe not. But if you trust yourself and you see that this is going to be something, then do it.
I would like to thank you, Professor Kellomäki, for this interview, and I think it's good advice for everyone. Trust yourself, follow your dreams, and do it.
Yep. Don't just dream, but do it. And thanks for inviting me. It was really a pleasure to come and have a chat about the RegJoints.